Discuss about the non-protein amino acids. Actually, these non-protein amino acids, although never found in proteins, perform several biological important functions. So, like non-protein amino acids and D-amino acids, speculated to before related autoimmune disease and to aging. First of all, understanding the role of non-protein amino acid and D-amino acids, autoimmune disease and aging, the determinations of these non-protein amino acids and D-amino acids is required. Any non-protein amino acids can be chemically incorporated into the majorly dissolves like incorporated into the peptides provided that appropriate methods are designed for protecting the functional groups. Non-protein amino acid with non-cytotoxic have been known to be incorporated into, for example, tyrosine and tryptophan residues in some proteins have substituted with M-fluorotyrosine and 4-fluorotryptophan respectively without any effects on the protein functions, and the 19F nuclei have been used like as a magnetic resonance. Some of the non-protein amino acids, some of these things in classified the various designs like this: alpha amino acids, non-alpha amino acids. Coming to the alpha amino acids, ornithine, citrulline, arginosuccinic acid, tyrosine and triiodotyrosine as adenosylmethionine, that is called SAM. Homocysteine 3,4-deoxyphenylalanine, dopa, creatinine and O-thiol azathioprine. These are the non-alpha amino acids: beta alanine and beta amino isobutyric acid and gamma amino butyric acid GABA and amino levulinic acid, that is ALA, and tharun. These are the non-amino acids of non-alpha amino acids. Coming to the alpha amino acids, ornithine. So ornithine, precursors of polyamine and hydrolytic cleavage of the guanidino group of arginine, catalyzed by the liver arginase and releasing urea. The other product, ornithine, re-enters liver's mitochondria and participates in additional rounds of the urea synthesis. Ornithine and lysine are potent inhibitors of arginase and complete like with the arginine. So arginine arginine also serves as the precursors of the potent muscle relaxant nitric oxide and in a calcium dependent reactions catalyzed by nitric oxide synthase. Citrulline is the intermediate in the biosynthesis of urea. L-Ornithine transcarbamoylase catalyzes transfer of the carbamoyl group of carbamoyl phosphate to ornithine, forming the citrulline and also orthophosphate. While the reactions occurs in the mitochondria matrix, both the formations of ornithine and the subsequent metabolism of citrulline take place in the cytosol. So entry ornithine into mitochondria and exodus of citrulline from mitochondria therefore involve the mitochondrial intermembrane transport systems. Arginosuccinic acid, arginosuccinic acid intermediates in the biosynthesis of urea. Arginosuccinate synthetase links aspartate citrulline via the amino groups aspartate and provide the second nitrogen of urea. The reactions requires the ATP and involve also intermediate formations of citrulline, cyclic AMP, also subsequent displacement of adenylyl monophosphate by the aspartate, then the forms of arginosuccinate. In addition to patients that lack of detectable arginosuccinate, the synthetase activity and a fold elevated for the citrulline has been reported in the resulting citrullinemia, plasma cerebrospinal fluid, that CSF. Citrulline levels are elevated and 1 to 2 citrulline are excreted in daily. Tyrosine and triiodotyrosine, we all know the TSH and T3 and T4, these are the thyroid hormones, some of the involves the epinephrine, non-epinephrine following the iodination hormones of T3 and T4. The reduction reductase, glutathione reductase, paradoxylase and deiodinase that converts the tyrosine to thyroid triiodothyronine. The clinical history, physical examinations and lab results were all consist with the primary hyperthyroidism. Accordingly, the patients was started an a large dose, like whenever low dose of thyroxine, it is important to begin therapy with a small dose like a T4, as large doses can be participated serious cardiac events due to the changes in the metabolism caused by the administration of hormones. Thyroxine, the free thyroxine, like this is the 4 picomoles per liter. S-adenosine methionine homocysteine, that is the SAM. S-adenosine methionine, the principal source of methyl group in the metabolism, contributes its carbon skeleton to biosynthesis of polyamines, speramine and spiramidine, homocystinuria, synthetase, cystathionine synthetase, lens dislocation and thrombotic vascular disease and mental retardation, osteoporosis, like autosomal recessive, homocystinuria, 5,10 methyl tetrahydroreductase and mental retardations, gait and psychiatric abnormalities, recurrent strokes, mental retardation, hypotonia and seizures and megaloblastic anemia. Pathways, enzymes and coenzymes involved in the homocystinuria methionine transferase, like a methyl group during its conversions of homocysteine. The defects in methyl transferase are in the subsequent metabolism of homocysteine by the pyridoxal phosphate B6 dependent cystathionic synthase, like increasing plasma methionine, and also homocysteine transferase, like via the methylation. These occur through the methionine synthase, reactions requiring the methyl cobalamin and folic acid. Deficiency in these enzymes, lack of cofactor, is associated with the decreased or normal methionine levels. Alternatively, pathway homocysteine can be remethyled beta 9, like homocysteine methyl transferase. Some of these things we will classify how the structure will, here it is remethylation, here remethylation to reductase, and also how the SAM is formation, like tetrahydrofolate reductase, here is some of the enzyme is required here, and also how the conversions of SAM, so methionine synthase to methionine reductase, here the methionine is here SAM, S-adenosine conversions of S-adenosine methionine, then also S-adenosine methionine, it will be involving conversions of by the involvement of methyl transferase in the formations of S-adenosine homocysteine. Then ultimately this is transferred to the homocysteine, is conversions of cystathione, also say homocysteine and serine. But here is homocysteine, again it is reutilized by the methionine, that by the enzyme here we required like betaine, like homocysteine methyl transferase. Also here is required some vitamin B12. This is the most important step here. Also that conversions of like tetrahydrofolate and methyl tetrahydrofolate and methyl tetrahydrofolate reductase. Here is we can folate reductase, some of the enzyme here it is inhibition of this step. So this is ultimately how the SAM and homocysteine formations, how the B12 and betaine, and also these are the enzymes here involving. This is the dopa dihydrophenylalanine. These are the importance of neurotransmitters and also neuromodulator, and this is the most important hormone here. So dopamine, non-epinephrine and epinephrine synthesis in the catecholamines, neurotransmitters. These the three are the synthesized in the common pathway, like amino acid L-tyrosine. So some of the dopa is a dopamine and neurons. Also this is the involving of dopamine hydroxylase. So how the synthesis of dopamine? So this is the L-tyrosine conversions of by the involving of tyrosine hydroxylase, the conversions of dopa. Dopa ultimately it is by the involving of dopa decarboxylase, involving the conversions of dopamine dihydroxyphenylalanine. So this is ultimately dopamine is non-epinephrine to ultimately epinephrine. Here is the SAM. We find out these things, SAM to SAM, S-adenosine methionine to it is conversions of methyl transferase S-adenosine homocysteine. So creatinine, we all know gum, that is glycine, arginine, methionine. These are the things we require. This is the tripeptide. We all know creatinine involving also muscle functions. Glycine, ultimately glycine, guanidinoacetin, creatine and creatine, phosphate and ultimately here creatinine. Kinase is a very important step here. Then glycine also. Okay.